All right, so I'm happy to be here and to present Cereno Scientific's vision, our significant progress the last year, and also our plans for the future. I hope you have some good questions for me when we're done. So our vision is to develop a first-in-class drug to prevent thrombosis. Thrombosis is the #1 killer on the planet. And there is a huge need for better efficacy and also to avoid the risk with the current therapies. Looking at who gets thrombosis and what the impact it has, so you can see that here is about half of the deaths are caused by cardiovascular diseases, and most of that is thrombosis related. So stroke in the brain, infarct in the heart, thrombosis in the veins. And here you see, so it affects the vital organs mostly. And you can see the background for thrombosis is the welfare diseases, so to speak: smoking, diabetes, overweight, inflammation, and so on. So it's very common, and it's the most common cause of death. Here you see, because it's so common, you can see it's a huge market for therapies. And it's a bit old data here, 2017, but around $20 billion per year. And it has a growth rate to double within 7 years. So by 24, it will be $40 billion. And that's the market we're going into. And we're going to hit the market about 24, 25 approximately with our drug. Now, what's used today? You probably know this, and it's called blood thinning drugs. Anticoagulants or antiplatelet drugs, two different kind of blood thinning drugs, so to speak. And the reason they're called blood thinning drugs is that they affect the coagulation system in the body. So it actually makes it harder for the body to coagulate if you have a bleed. It prevents thrombosis pretty well, but it could be better. But the major problem here is that you see the man here is red, all the vessels are red, and it's to illustrate that if you take these drugs, and you take them every day, most people for life, after an infarct or after you've had a stroke. The problem is that you're at risk of a bleed, a major bleed or any kind of bleed 24/7. So every minute or hour you're on these drugs, you have a risk to experience a bleed. And it can be a fatal bleed, it can be a major bleed, or it can be any kind of bleed. For instance, nosebleed that doesn't stop. You have to go to the hospital. So all drugs act as blood thinning drugs, and it's a systemic drug effect. Now, it increased the risk of bleeding, as I mentioned, and the problem with that is then that the pharmaceutical industry has developed these drugs in lower dosages than would have been the case if they didn't cause bleed. So if you increase the dose, it's high risk for bleedings. So there's both an efficacy problem on the market and a risk problem, adverse reaction problem. So there is a need for new preventive strategies on the market. Okay. How common is this? Well, major bleeds are, and this is in clinical material, up to 2.5–3% get major bleeds, dangerous bleeds in stomach, brain, and up to 1/4 in clinical materials get any kind of bleed. So every 4th patient. And this is in clinical material where the most risk-prone individuals are not included. So, big problem. Now, this is to illustrate the body, and the body has two different ways of dealing with bleedings in the body. So you see to the left, there's a coagulation system, so the body can coagulate. So you stop a bleeding on the skin, for instance, you can see it. It becomes a little coagulation and a scar. And the body also has another system called the fibrinolytic system. So we are equipped with a system to dissolve blood clots. All right, Thank you. So we are equipped with that system like a high-rise that has sprinklers. You know, if there's a fire in a room, the sprinkler in that room goes on and water comes out. That's how our defense mechanism works in the body, and I'll illustrate that. And if you are a risk patient, that doesn't work. Now, as I mentioned, all the drugs on the market are blood thinning. They act on weakening the coagulation system to prevent thrombosis. We are acting here on the fibrinolytic system. So we are restoring that system to become better, a better defense than risk individuals have. That system is too weak. And let me show you how it works. So this is a vessel. It's a plaque, atherosclerotic plaque that most of us have in the room here, I think. And when you get older, they can burst, and then you get the bleeding inside the vessel. And when that heals, you get a blood clot forming, and you don't want it to occlude the blood flow. When that bursts, there is a signal going up to the cell wall inside, and it releases this fibrinolytic water, so to speak. It's a protein, and it dissolves the blood clot so it doesn't occlude, grow and become too big and occlude the blood flow. This is a very important safety mechanism. There is another protein in the blood floating around called pi, and the main activity of pi is to block the action of TPA that's coming out. It's a balance system. It's all good if it works, but in risk individuals, the TPA, the sprinkler, there's no water in it, so to speak. There's too little of the protein, and there's too much of the floating pi. So you get too little coming out, and what's coming out is blocked by pi. And then the blood clot can grow, and you get an occlusion, and you have an infarct. So that's very bad. So these are two risk factors for myocardial infarction and stroke. Just very quickly look at pi here, the floating protein. That's very high in the morning hours with risk patients, and that's exactly when it's most experienced to have stroke or infarcts, in the morning hours. And you can see that to the right on the black staples there, angina pectoris is a coronary event, and there's acute myocardial infarction. You can see the hours correspond with the early morning hours, somewhere between 5 and 9 in the morning. So that's when you want your defense mechanism to be intact. And look here at this slide. This is a heart and a coronary vessel, and you see to the left, you see it's working. So you get a small bleed, TPA is showered out, the sprinkler, dissolving the clot so it doesn't become too big, and pi blocks the rest of TPA and takes it away, and it's all controlled. If you look to the right here, you see a risk individual. There is very little TPA in the cell wall. There is a bleeding starting, and the blood clot is growing, and pi is in high concentrations. Let's say this is in the morning hour, and you get an infarct. The defense mechanism is too weak. And as you can see from the heart, downstream there, it's all white. You see the heart, that's necrosis, that's dead heart cells. And that's what happened in a myocardial infarction. So you either die or you get an incapacitated heart. You can develop heart failure and so on. So you want your defense mechanism to be intact. What we are developing is a product, CS1 we call it. It's our lead project. And that will make possible a restoration of the defense mechanism. And I'll show you how. So we will increase the synthesis, the storage of TPA and the release of TPA, and will also reduce pi in the morning hours. And I'll show you the documentation we have. And that way, if you take this in advance every day, you will have a defense mechanism that's stronger. So we have a dual mechanism of action. Our product can increase the synthesis, storage, and the release of TPA from the cell walls, and we at the same time reduce the risk factor pi from the bloodstream in its concentration. And by that we have a stronger defense mechanism. And the beauty with this, if you look to the right in the picture with the arrow, It's activated only when and where the body needs it. The body itself activates this. So we are not causing a situation where you have an individual that are at risk of bleeding 24/7. So it's a very different approach compared to what's out there today. Yeah, so the advantage with this first-in-class drug, CS1, is that we prepare the body to have a good defense mechanism against an event. And we restore the fibrinolytic capacity, and it's activated only when and where needed. So you see this man here to the left, or woman, is not all red. It doesn't have a risk of bleeds 24/7. So our objective is now to develop this drug to use it as a preventive therapy. And we think it's going to be very valuable as an alternative to the existing drugs out there. It's going to be safer and it's going to be effective. And the major indications are deep venous thrombosis, myocardial infarction, and stroke. And prevention of stroke is the major indication here. Now let me just quickly go through some of the data that we have to support our vision and our development program. So we have in vitro studies, so cultured human endothelial cells. We have animal models in mouse and pig. I'll show you a mouse model today and the data. We have human physiological data, and I'll show you that. And there's also very interesting epidemiological studies indicating that this really works in man already. And then we have the data on our phase 1 study that we concluded last summer with very positive data. So first, the in vivo data on cultured cells. And here you have two pictures. One is without treatment and the other one is with the active substance that is in our drug, CS1, valproic acid. And you can see the green stains here are actually TPA. So TPA in the cell wall. And remember that I mentioned that that's one of the effects, is that if you take this in advance, there will be much more production of the protein that you need to have in the cell wall. And the green stain is staining that protein. You can compare the pictures and you can see there's much more. This is our proof of concept data in live animals treated for 3 days, 72 hours. And you can see here on the x-axis is time and on the y-axis is the fibrinogen content. And these mice were hurt in a vessel, a vein, and they were pre-treated and hurt, and then there is a bleed and there's a thrombosis formation. And the treated group is the red line and the control group is the black line. And you can see already after 2 minutes, in the very beginning there is a significant difference between the curves. So there's much less thrombosis buildup in the mouse that was pre-treated. So it has a defense mechanism already against infarct, if you will. And the photograph shows the size of the thrombosis in the two, treated and pre-treated. And you can see it's much less in the lower picture. Now let's move on to experimental studies in man, and here are 2 pictures, and they show that the substance in our drug has an impact on the 2 biomarkers for infarction or thrombosis. The left picture is a forearm model treated over 4 weeks, and it was documented that we release TPA better if you are pre-treated. It's the top line. So there's more TPA released in the humans. And they were people with coronary disease or heart disease. On the picture to the right, there is an effect, a study on over 2 weeks to show the impact on the other important protein here, the risk factor for thrombosis, pi, the one that was floating around in the body. And here we saw up to 50% reduction of pi in blood. So we have documented in man impact on these 2 markers. Now let's move on to epidemiological studies. There are actually 3 different publications. There are 4 here, but there are 3 different studies. One Danish in 2011, another Irish that confirmed the first one in 2014, and one last year, 2018. And it's research groups that have looked backwards into patients that have epilepsy. And the reason they're looking at that is that the substance that we have found is already used for epilepsy, has been used for 45 years as a drug for epilepsy. So they have gone backwards and looking at, does it matter if what kind of drug you are on for cardiovascular incidents, myocardial infarction and so on. And all of these studies has confirmed the indication that it matters. So if you are on this substance, VPA, which is an R drug, you have less infarcts and you have less stroke. So already in long-term studies, and I'll show you one because we don't have time for all. This is the Danish study published in 2012, 150,000 patients were follow-up with epilepsy. There were a variety of drugs. One of them were VPA, used for epilepsy, 10 years follow-up. And the difference in myocardial infarction in the data material was almost 40% less myocardial infarction, only in the group that had VPA. Similar kind of data is for stroke in the latest study from 2018. So it's very encouraging that this data already exists in man. Now, let's move on. So we have developed this VPA into a very specific formulation. It's an innovative concept where we have a delayed immediate release formulation of VPA. So if you take the tablet, it prolongs the release of the drug, and then it releases it. And the reason we've done that is that we want to affect pi in the morning hours, as we spoke about before, as I mentioned. At the same time, we want to have an effect on the TPA production. And the data was, we published the data, presented the data in June last year. And no problem with safety and tolerance, and we didn't expect it because the drug is already used in man, the substance. We were looking for these pharmacokinetics with a specific profile in the blood so we could affect pi the most. And we actually found that pharmacokinetics well. We used 2 different dosages. And what's not normally done in phase 1 trials is that you look for anything else than safety and tolerance. But we looked for an impact on pi, and we documented a significant reduction of pi in the morning hours with CS1. In addition, we also documented bleedings, and there were no increase in bleeding in this study. So that was very encouraging, and we had already 8 months previous to that started to do a procurement to find a CRO to do our phase 2 program. And we changed our strategy from doing a small phase 2 program to doing a really big, I would say, phase 2 program for a company our size. And the reason is that we want to do it the same way that the big pharma has done proof of concept in man. The products that are now on the market selling for most part of the $20 billion US have done this kind of study that we will do. So we signed a contract with OCT. So we will do a study in circus 30 centers, multi-center in Russia and Bulgaria. And we'll do it in orthoplastic surgery, actually total knee replacement. And the reason is that when you go through that kind of surgery, the risk of thrombosis, blood clot, is increased. Other good news during the fall here was that we got a major good news. We got patent approved in the US, which makes up about 50% of the market. So that was very positive. Other news that we got was, or what has happened recently, is that we have terminated our convertible bond financing that we signed a year ago. And we are pursuing other financing. I'll come to that at the end. We also got a patent granted in Australia this spring, another important market. And then we have established a a very strong scientific advisory board. I'll mention it at the end here. We're working very actively at the moment. A couple of other events. We acquired a new substance from a group at AstraZeneca. Used to be at AstraZeneca, but they're at the BioHub, where we are also located in Gothenburg, AstraZeneca's facilities. Emeriti Bio. They are the group that were behind Losec and Losec's follow-up, Nexium, Brilic, Plendil. Those are the chemists, and they have formed this company. And we have now acquired one substance from them that fits very well with our concept. This was done in March, and we filed quite rapidly a patent here in May on that compound. Yeah, so I will jump this. So we are developing a new preventive therapy for thrombosis. Reasonably short time to market because it's been in man already, the substance. We have a double effect, TPA synthesis, reduction of pi in the blood. We have documented this in vitro, in vivo, experimental human studies, epidemiological studies, 3 of them. We have a positive phase 1, and now we're pursuing phase 2. We got patent in US and Australia, and we have expanded our pipeline. Briefly mention something about scientific advisory board. These are heavy cardiologists and researchers. I'm going to meet with them in Athens this week in connection with the European Society of Cardiology's annual heart failure meeting. And so Dr. Bertram Pitt is chairman. I've worked with him in the past in major studies. And Dr. Gordon Williams is from Harvard Medical School, so is Deepak Bhatt. Professor Faiz Sanad is from France, runs the cardiovascular clinical trial program that FDA used to run in Washington every year. And Gunnar Olsson has been one of the heads of AstraZeneca's research for many years, both cardiovascular and gastro. Now, upcoming milestones. So we are imminently going to submit our phase 2 study application to authorities for approval. We thought we were going to do it in Q3, but we've moved it backwards to Q2 because it seems that we are ready. So look out for that news. We are pursuing a share issue very shortly. I'll mention that at the end. And so we expect the authorities to approve our phase 2 during the fall. We hope so. And then we are scaling up the production of our clinical material, including our drug, for that study. And that's a hefty job to do. And we're actually working at it at the moment. And we expect to start the study during the spring and to complete it by the end of 2021. So the study takes about a year to do, and then there's another 6 months before and after. So end of 21. That's it.
Thank you very much, Sten. Very interesting to see all the things that you have accomplished the last year. Preparing for a phase 2 study. You said that this study is designed or is going to be made the way a big pharma would do it. What's the reasoning behind that? Because I imagine that makes it a little bit more expensive than it could have.
Absolutely. So yeah, it's if you have big visions, you have to do the job. So and we have, we're very skilled people in our group and external advisors, and we adjusted our strategy to do it the way Big Pharma does it, because we don't want the data to be questioned, and we want to make the best effort possible to document proof of concept in man to prevent thrombosis. So that's why, and it is a bigger price tag. So yeah.
But you gain some in the end, hopefully.
Absolutely, we hope so. Yeah.
So in the end, you mentioned the new scientific advisory board. Like you said, there are quite distinguished names on the board. What has attracted these people to Cereno, you think?
Well, firstly, I have started a couple of initiatives in the past that's been going very well. Survival trial in heart failure and a new concept with aldosterone in the 90s. And Dr. Pitt, who's chairman of the Scientific Advisory Board, was actually the chairman of that survival trial, which was published in 99. So that was went very well. And Fayez Sanad was also in that study, and in the next study, which was started together with colleagues when I joined AstraZeneca, I initiated with them a survival trial with Astra's beta blocker, Seloken or Toprol XL. And these two studies were published the same year, and they were very positive. So now when they got to know what we were doing here, these are very busy people, and they go for science. And actually, they wanted to work with Cereno.
Yeah.
So yeah, so when they got to know what we were doing, and you will later see the results of our work together. We formed this scientific advisory board this spring, and we're meeting now. But we've already started working, and I will be happy to share more of that in the coming months with you, what we're doing.
Yeah, great. We are running out of time, so I'm going to let one question from the audience.
Is your substance CS1 identical with valproic acid, or is it a modified version that has implications for the studies you have to do and the size of the studies?
Yeah, exactly. So the substance that we have in our drug, CS1, the chemical substance is identical. So what we have done with that is we have reformulated. It's quite complicated formulation that we have developed together with Galenica since late 2006. Beginning 2016. So it's the same substance. So it's the same substance that you see in the epidemiological studies and so on, but different dosages and very different formulation. So the plasma profile of it and the concentration in the body is very different. And the reason is that if you give this for epilepsy, you need to have certain concentration to prevent the grand mal or whatever is happening in epilepsy. But in this indication, what we are after is stimulating it's an epigenetic stimulation of TPA synthesis. That takes some time to get going, and then there's a constant reduction of pi in the blood that we need to get done. And for that we need different dosages and different pharmacokinetic profile. And that's what we have developed. So we have 3 patent families. So we have filed usage patent first, and then a patent in relation to pi and thrombosis, and then a patent in relation to the formulation. And we are in national phase with the second patent family in 21 countries at the moment.
Because it has implications for the safety. Now, the phase 2, how many patients are you expecting in that? And the following question, how many would you think are needed for a phase 3?
Well, yeah, so I can't really tell you how many we have in this study yet, because I haven't published it yet. We haven't released that information. And it depends also in relation to our discussions with authorities on a clinical protocol that we are submitting. But it's 30 centers, and it will be several hundreds patients that we are pursuing. And you're very right. I think the safety for new chemical entities, you never know what's going to happen. So if you do small studies in the beginning, you might get hit by a side effect in phase 3 in a major study, or even in phase 4, like COX-2 inhibitors, for instance. They were pulled from the market. In our case, this has been in man for 45 years. So it's quite well known. It's been in much higher dosages than we will use in our drug indication. So we expect less documentation needs and absolutely much less risk. And so again, several hundred patients in these studies. And in the phase 3 studies, I have to return to you with that. We have been looking at it. So it's not small studies. All right.
So we will be looking forward to the release of more information once you have sent in the application for the phase 2 study to regulatory authorities. Yeah.
I have one more thing I'd like to say, but
yeah, we can do that in a little while.
Yeah.
First, I'm going to thank everyone for watching in the US, and thank you everyone for coming. Nu byter jag till svenska. Sten har en slide till som han gärna vill visa.
Det är ingen surprise kanske, men vi terminerade ju vår financing agreement för fonden som hjälpte oss fram till där vi är idag. Och nu har vi precis releasat vi vårt prospekt igår för en share issue, en emission som vi 23 maj till den 7 juni så är det teckningstid för den, och den är ju till för så att säga de som har aktier redan. Men 2,90 är teckningskursen och vi planerar att ta in 55,6 miljoner. Den är garanterad till 80% och management har puttat in 6,5%, 3,6 miljoner. Där är en overallotment också, om det skulle vara många som tecknar sig så tar vi in 5 miljoner till. Och detta är för att ta oss fram till och med start av fas 2-studien. Sen behöver vi göra en finansiering till för att genomföra hela fas 2, för den kostar så pass. Den här ekonomin är också till för att driva patent och andra saker, men också för utvecklingen av CS014 och nästa substans som är i preklinisk fas, den jag berättade om. Så har ni aktier så teckna gärna. Ni var med på resan. 3
snabba argument för varför man ska vara med på den här resan fortsatt.
Ja, alltså ett argument är ju, huvudargumentet tycker jag är om man verkligen tror på teknologin. Jag är en teknologiperson, jag går på teknologi. Den andra kan ju vara mer finansiell, alltså att ett bolag som är i vår fas är normalt sett, man ska inte prata för mycket om värdering, men normalt sett hyfsat värderat på börsen. Vi är värderat till 60 miljoner kronor ungefär. Och vårt börsvärde sjönk med 80% under den här finansieringen med fonden som var convertible bond, som tryckte nya aktier och sådär var 6:e vecka och släppte ut på marknaden. Och den har vi då terminerat, så att jag tror att vi har ett nerpressat börsvärde idag på 60 miljoner. Det skulle varit kanske 200-300 i nuläget. Och den tredje är ju att om man ska man ska ju helst in innan en fas 2 görs, för när den blir färdig, om den är positiv, så lär det få ringa på vattnet så att säga.
Nyckelord, om den är positiv.
Ja, absolut. Så är det ju med all forskning.
Kan du säga någonting bara om den här CSO14? Vad är det för någonting?
Nej, inte mer än att den är innovativ. Faktiskt. Så att det är inte så mycket än. Och det finns flera skäl till det. Det är patentrelaterade skäl och det är också konkurrensskäl. Så att vi avvaktar lite med det, tyvärr får jag säga så, denna gången. Men vi hoppas komma tillbaks med lite information kring den om möjligt här under hösten. Men det är en spännande substans, absolut.